The overall goal of the following pipeline is to better understand aging and dementia by obtaining MRI-derived measures of regional brain atrophy and neuroimaging biomarkers of small vessel disease. This is achieved by first generating a skull-stripped brain mask and a basic brain tissue segmentation to accurately quantify the total intracranial volumes. As a second step, a tri-feature lesion segmentation is completed to quantify and classify lesion subtypes for accurate imaging biomarker measurements of small vessel disease. Next, anatomical landmarking is performed on each individual to parcellate the brain into 26 standardized regions of interest. Results are obtained that show differential regional neurodegeneration and vasculopathy patterns between disease groups based on brain tissue atrophy measures and lesion volumetrics obtained from structural MRI.
Our analysis approach can help address key questions in brain imaging of aging and dementia. The personalized pipeline we have developed allows researchers and clinicians alike to better understand diagnosis, progression, and treatment response in individual people, as well as different patient groups. It is especially useful for people who have cerebral small vessel disease in combination with neurodegenerative disorders. This is very common in the elderly, especially in Alzheimer's disease. The pipeline can also be applied to people with other neurological disorders, such as vascular dementia and stroke, frontotemporal dementia, traumatic brain injury, and multiple sclerosis.
Visual demonstration of this method is critical as the manual interventions